Imagine having a universal map of the human body.
Mm-hmm.
But I am not talking about the actual anatomical body you're walking around in today. I mean a map of how to grow miniature, living, breathing versions of your own internal organs from scratch, right there in a petri dish.
Yeah, it sounds completely sci-fi, but we are genuinely looking at the bleeding edge of biological modeling right now.
It is wild.
It really is. I mean, we are aggressively moving away from studying, static, flat cells on a glass slide. We are moving into this era of building dynamic, three-dimensional living tissue.
And that is exactly what we are jumping into for this deep dive. We have our hands on this monumental paper from the journal Nature Genetics. It was published in May 2025.
Right.
And it is titled, let me get this right, An Integrated Transcriptomic Cell Atlas of Human Endoderm-Derived Organoids. The scientific community is just calling it the HEOCA.
Which is much easier to say, thankfully.
Definitely. So what we are doing today for you listening is decoding this atlas. We're going to figure out how scientists managed to synthesize and map nearly 1 million individual cells from 55 different publications, and why this is just a massive tectonic shift for modeling human disease without, relying on animal models.
The HEOCA dataset really is a complete paradigm shift, because for years, the whole field of organoid research was just incredibly fragmented.
Like the Wild West.
Exactly. You had these isolated, highly variable lab experiments happening all over the world. And this paper basically brings all of that chaos into a unified, standardized ecosystem.
Right. And we're going to walk you through exactly how they did it. We will unpack the mechanisms, the biological hacks they use to actually force these mini organs to mature, and look at what happens when you intentionally infect these lab-grown organs to study human sickness.
It gets intense.
It really does. But to understand the solution, we have to understand the chaos that came before it. We have to look at this wild west of mini organs. Now, we're talking specifically about endoderm-derived organoids. And the endoderm is basically the foundational canvas of our internal plumbing, right?
Essentially, yes. So in a developing embryo, the endoderm is the innermost germ layer.
Okay.
It is the cellular foundation that eventually develops into the highly specialized linings of your most vital internal systems. So we are talking about your lungs, your thyroid, the pancreas, the liver, your stomach, and your intestines.
That's a lot of major plumbing.
Right, and modeling these linings is notoriously difficult because they act as the complex interface between your internal biology and the outside world. I mean, they process literally everything you ingest and inhale.
So to grow these structures in a lab, researchers use stem cells. And looking at the source material, there are 3 primary starting materials. You have PSCs, which are pluripotent stem cells. You have FSCs, which are fetal stem cells. And ASCs, adult stem cells. But here's where I get hung up.
Okay, what is the hang-up?
Well, if a lab in Tokyo and a lab in Boston both order a batch of pluripotent stem cells and they throw them into a nutrient broth with a soup of growth factors to try to grow a mini gut,
right?
How do they know what they just grew actually acts like a human gut? Isn't it essentially just a random clump of confused cells trying their best to follow vague chemical instructions?
You are hitting on the exact crisis that has plagued stem cell biology for the last decade.
Really?
Yeah, it is known as the fidelity problem. Fidelity just refers to how faithful or accurate the lab-grown cell is to the real living cell inside a human body. Because protocols vary so wildly from lab to lab, the outcomes are incredibly inconsistent. One lab uses a slightly different temperature, or another uses a different nutrient broth, or they handle the cells for an extra 2 days.
Just tiny variations.
Exactly. And as a result, assessing the true biological fidelity of these models was almost impossible.
And reading through this research, the consequences of that inconsistency are just wild to me, because pluripotent stem cells, those PSCs, they have the potential to become literally any tissue in the human body.
Right. They are a blank slate.
So they are desperately searching for signals to tell them what to do. If the chemical signals in the petri dish are even slightly off, they drift. Like you could be trying to grow a liver organoid, and because the cells get confused without the exact spatial cues of a real womb, you accidentally grow a few stray brain cells inside your liver model.
Which completely ruins the model, especially if you are trying to test, say, a liver toxicity drug. We call those off-target cells.
Off-target, right.
Yeah. Pluripotent cells are exquisitely sensitive to their microenvironment. Without the precise mechanical pressure, the blood flow, and the chemical gradients of a living embryo, they just wander down the wrong developmental pathways.
That makes total sense.
So you end up with these off-target cells, and on top of that, you get massive variations in cellular maturation. Some cells in the dish act like they belong to a fetus, while others are slightly more mature. It creates this chaotic, noisy data set.
Okay, so let's do a quick recap for you listening to make sure we are all on the same page. Point 1, organoids are 3D mini organs grown in a dish. Point 2, endoderm organoids model our internal linings, like the lungs, gut, and liver.
Hold on.
And point 3, the big challenge is fidelity, making sure lab-grown cells actually match real human biology and aren't just confused off-target cells.
Exactly.
So question for you listening. If you were a scientist, how would you prove your lab-grown cell is exactly the same as a cell inside a living human? Hold that thought, because we are about to look at the ultimate answer to that.
And this brings us right to the core of the Nature Genetics paper. The researchers behind the HEOCA realized that to solve this fidelity problem, they could not just run another isolated experiment.
Right.
They had to build the Rosetta Stone of cellular biology. And the scale here is just staggering.
It really is. To create this centralized reference map, they didn't just grow their own cells, they integrated existing data from 218 different biological samples covering 9 different human organs. In total, they performed an analysis on 806,646 individual cells.
Nearly 1 million distinct cells mapped out. But the mechanism of how they actually read those cells is what is so fascinating. They use cRNA-seq.
Which stands for single-cell RNA sequencing.
Right. Let's break down how this actually works, because it is not just looking at a cell under a microscope.
Not at all.
Under a microscope, many cells look completely identical. To truly understand a cell, you have to look at what it is actively doing. Inside every single cell in your body, the DNA is the permanent, locked instruction manual.
Like the master blueprint.
Exactly. It contains the code for everything. But RNA is the temporary, active copy of the specific instructions being used at that exact millisecond. Single-cell RNA sequencing captures and reads those active mRNA transcripts.
So it is essentially scanning the exact pages of the manual the cell happens to have open on its desk at that moment.
That is a perfect way to put it. By sequencing the RNA of a single cell, scientists are reading a highly detailed barcode. It doesn't just tell them the cell's basic identity, like whether it is a lung cell or a gut cell.
Right.
It tells them its exact biological state. Is it a mature cell? Is it actively dividing? Is it under stress? Is it preparing to defend against a virus? The cRNA-seq technology captures that entire functional landscape.
That is incredible. But here is the massive logistical hurdle the paper highlights. They collected the sequence data from 55 different published studies.
Yeah, that is a lot of different sources.
Right, which means 55 different labs using entirely different brands of sequencing machines, different chemical reagents, and different tissue handling times. It feels like trying to combine 55 different books into one cohesive manuscript. But every single chapter is written in a different language, printed in a different font, and formatted completely differently. If you just dump that into a database, it is going to be unreadable.
In bioinformatics, that specific problem is known as a batch effect.
Batch effect, okay.
It's a massive roadblock. Technical noise completely obscures the underlying biological truth. If you take those 800,000 cells and put them into a standard computational model, the cells won't actually group together based on their biological function.
Wait, really? How do they group then?
Well, all the cells sequenced in Boston will cluster together, and all the cells sequenced in Tokyo will cluster together, simply because the sequencing machines in those labs had slightly different baseline calibrations.
Which completely defeats the purpose of building a universal map.
Exactly.
So how do you strip away the font and the formatting to just read the actual text?
The researchers had to test 12 different computational integration algorithms to solve this. They ultimately selected a deep learning model called SCIPI-COLI.
Scully.
Right. This algorithm is essentially an advanced data harmonizer. It works by projecting all the single-cell data into a shared latent space. It learns the technical variations, the batch effects from the different labs, and mathematically subtracts them.
Oh, wow.
While carefully preserving the true biological variants.
So Scopoli acts as the ultimate universal translator. It filters out the noise of the laboratory equipment and leaves only the pure genetic barcode of the cell.
Exactly. And once they ran nearly 1 million cells through this algorithm, the resulting taxonomy was incredibly clean. They categorized this massive sea of data into 5 main cell classes: 48 distinct cell types, and 51 highly specific subtypes.
And this is where the map starts revealing things we could never really see before.
Yeah.
When you have a harmonized database of this scale, you can spot conserved biological patterns across totally different organs. For example, the atlas revealed the universal deployment of goblet cells.
Right, the goblet cell.
Yeah. These are highly specialized cells responsible for secreting mucins, which form our protective mucus layer. The HEOCA data showed that goblet cells made up about 68% of the cellular population in intestine models and about 32% in lung models.
Which, from a biological standpoint, makes perfect sense. Both your respiratory tract and your digestive tract are constantly exposed to foreign particles and pathogens.
Right.
They both desperately need that sticky, protective mucus barrier to trap invaders.
Exactly. The map also tracked basal cells, which serve as foundational progenitor cells. The data proved that these basal architectures are universally distributed across models of the lungs, the salivary glands, the intestines, and the thyroids.
It is fascinating.
It shows us that the endoderm uses a shared toolkit of cellular building blocks, regardless of which final organ it is constructing.
It really highlights the elegance of human biology.
Okay, time for another quick recap. Point 1. HEOCA is this integrated database of over 800,000 single cells. Point 2, single-cell RNA sequencing acts as a cellular barcode reader, showing us what the cell is actively doing.
Yes.
And point 3, the deep learning algorithm Scapoli was used to eliminate all those lab-to-lab batch effects so the data could actually be compared.
Perfect summary.
So think about this for a second. If you had access to this standardized map, what is the first thing you would test against it? Because the obvious next step for these researchers was to use this map to grade the actual organoids they were growing.
To see if they were actually any good.
Exactly. Let's look at the specific case studies the paper highlights, starting with the human intestine, which they call the HIOCA, and the human lung, the HLOCA.
The findings regarding stem cell maturation in the intestine are just profound.
How so?
Well, the researchers compared the laboratory-grown intestine organoids against a primary reference atlas. This primary atlas was built using data from actual human tissue biopsies, covering both adult and fetal stages.
Okay, so a real-world baseline.
Right. They wanted to know which starting material actually produces the best adult gut in a lab.
And looking at the data, the adult stem cells, the ASCs, were the clear winner here.
By a massive margin. Organoids derived from adult stem cells were remarkably accurate to adult human biology. They hit a 98% on-target rate.
Wow, 98%.
Yeah, they properly formed the complex cellular crypts and villi you expect to see in a mature, functioning human intestine.
But the pluripotent stem cells, the PSCs, did not fare so well. According to the research, the PSC-derived organoids were essentially stuck in a developmental loop.
Yeah, they hit a wall.
They looked far more like fetal tissue than adult tissue. They completely lacked the complex cellular diversity needed to model adult digestion or adult disease.
Exactly.
So if I am a clinical researcher and my entire grant is focused on studying an adult-onset disease like, say, colorectal cancer, starting my organoids with pluripotent stem cells seems like a total dead end. The cells simply haven't grown up yet.
That was the fear for a long time. And it highlights the limitation of an isolated petri dish. But the HEOCA data validated an extraordinary biological workaround called xenografting.
Xenografting. This is the biological hack.
It is. Researchers took these early-stage, fetal-like PSC organoids that were basically stalled out in the dish, and they surgically implanted them into a living mouse host.
That is wild.
They used the mouse's body as a living, breathing incubator.
I want to pause on this mechanism because it is just fascinating. The cells in the dish have the exact same DNA as an adult cell. They have the genetic blueprint to become mature adult cells. But sitting in a plastic dish with some chemical nutrients just isn't enough to trigger the instructions.
Right.
It is like having the blueprints for a skyscraper, but no construction crew to actually read them.
That is exactly the issue. A plastic dish cannot replicate the sheer dynamic complexity of living organism. When you implant that organoid into a mouse, you are suddenly introducing vascularization.
Blood.
Exactly. The mini-organ gets hooked up to a real blood supply. It is subjected to systemic hormonal signals from the mouse's endocrine system. It experiences rhythmic mechanical stretching from surrounding tissues. It interacts with an active immune system.
And those invisible systemic forces are the missing cues, because when the researchers sequenced the cell after they had matured inside the mouse, the transformation was staggering.
It was a night and day difference.
The organoids had unlocked their adult genetic pathways. They developed highly specialized adult cells that were totally absent in the Petri dish. They started producing Paneth cells, which are crucial for secreting antimicrobial peptides to control gut bacteria, and tuft cells, which are sensory cells that detect parasitic infections.
It proves that genetic potential is heavily dependent on environmental context. You can see this exact same principle mirrored in the human lung organoid cell atlas too, the HEOCA.
Oh, right. Let's talk about the lung data.
The lung data definitively proved that the initial recipe, the stem cell source you choose, strictly dictates your structural outcome.
It is so clear in the paper. It is like choosing your construction materials. If you start with pluripotent stem cells, you are essentially working with raw, unformed clay.
Right.
You have maximum potential, but the resulting lung organoids heavily express early embryonic markers like FABP1 and AFP. They are just early-stage progenitor cells. They don't have the specialized structures to actually function as a mature lung.
Conversely, when researchers used adult stem cells, which we can think of as highly specialized tools that already know their specific job, the resulting organoids reliably produced a high proportion of mature club cells.
Club cells, okay.
Yeah, club cells are essential in the adult body. They secrete proteins that protect the lining of the bronchioles from inflammation and toxic damage.
And if you use fetal stem cells, you hit this middle ground, reliably producing those mucus-secreting goblet cells and neuroendocrine cells.
Exactly.
So the map basically eliminates the guesswork. If you want to study a specific part of the lung, you consult the atlas, pick the precise stem cell recipe, and you know exactly what level of maturation you are going to get.
It creates a predictable engineering framework for biology. We now understand that adult stem cells yield highly accurate adult tissues, while pluripotent cells require the complex systemic environment of a living host through xenografting to push past their fetal state.
Think about what that reveals about your own body for a second. We take it for granted, but every single cell functioning inside of you right now relies on an incredibly delicate, invisible symphony of blood pressure, oxygen gradients, and hormone signaling just to maintain its identity.
It is true.
Take those forces away, and the cell literally forgets how to act like an adult.
It is a humbling reminder that biology is deeply, deeply interconnected.
All right, time for recap number 3. Point 1, adult stem cells make the most accurate adult organoids right out of the gate. Point 2, pluripotent stem cells default to fetal states in a dish. And point 3, xenografting using a living host like a mouse forces those fetal-like cells to finally mature into adult cells.
Nailed it.
So, knowing that a lab environment isn't enough to fully mature a cell without a biological host, what does that tell you about the mysterious, invisible forces inside a living body?
It is a lot to think about.
It really is. So we have built the map, we know how to grow the healthy cells, and we know exactly how mature they are. The final phase of this paper is where we put all of this to the test. What happens when we take our perfect, healthy mini organ and subject it to severe biological trauma?
Ah, yes. In functional genomics, we refer to this as a perturbation experiment.
Perturbation.
Right. It's the ultimate stress test for fidelity. The researchers needed to know if these lab-grown models actually react to disease the same way real human tissue does.
Makes sense.
To do this, they selected highly accurate ASE-derived ileum organoids. The ileum is the final section of the small intestine, and they subjected these mini intestines to 2 very different, highly aggressive attacks.
The first attack was a simulated viral infection. But they didn't use a live virus, right? They flooded the organoids with interferons, specifically IFN-alpha and IFN-beta. What exactly is the mechanism of an interferon attack?
Interferons are rapid response signaling proteins. In a real human body, when a cell is breached by a virus, it immediately synthesizes and releases interferons into the surrounding tissue.
Like an SOS.
Exactly. It is a biochemical distress signal, telling all the neighboring cells to immediately halt their normal functions and raise their antiviral defenses.
Wow.
By artificially flooding the organoids with these interferons, the researchers triggered a massive systemic panic response within the tissue, perfectly simulating a severe viral invasion.
And the second attack was acute pathogenic inflammation. They hit a different set of organoids with a brutal cocktail of inflammatory cytokines. We are talking tumor necrosis factor, or TNF, interleukin-6, and oncostatin M. This is basically the biological equivalent of setting off a localized fire alarm.
Precisely. They let these attacks rage for 24 hours. And then they ran the cells through single-cell RNA sequencing to read their active genetic barcodes.
To see what manual pages were open.
Exactly. They compared the stressed dying cells against the healthy baseline map of the HEOCA. The differential expression, the genetic fingerprint of the trauma, was incredibly clear and distinct for each type of attack.
Let's look at the viral response first. The data showed that the interferon attack activated a highly specific set of defensive genes, most notably ISG15 and OAS1 through 3. I want to dig into ISG15 because its mechanism is just brutal.
It really is.
When that gene turns on, it regulates a process called autophagy.
Yeah, autophagy translates literally to self-eating.
Self-eating.
Right. It is a survival mechanism. When the ISG15 gene is highly expressed, the cell actively begins quarantining and digesting its own infected organelles and cellular structures.
Oh, man.
It destroys its own compromised internal machinery to prevent the virus from using it to replicate. It is a total scorched earth defense strategy.
That is terrifying but amazing. And what about the inflammation attack? The cytokine cocktail produced a completely different genetic fingerprint, right?
It did. The inflammation attack highly activated a different cluster of genes, primarily LCN2 and IL-32. These genes don't trigger autophagy. Instead, they regulate the cellular response to chemokines.
Chemokines.
Yeah. Essentially, the cells were furiously producing signaling molecules designed to attract circulating immune cells to the site of the damage. They were sending out a beacon for white blood cells to come and fight the inflammation.
But here is the absolute crux of this entire deep dive. It is an impressive parlor trick to make a clump of cells in a plastic dish react to a chemical. But does that isolated reaction actually have clinical relevance?
That is the million-dollar question.
Did the genetic panic in the Petri dish look anything like the actual suffering of a human patient?
This is where the paper delivers its most profound evidence. The researchers didn't just guess. They took the exact genetic fingerprints of the inflamed, stressed organoids and cross-referenced them against clinical biopsy data.
From real people?
Yes. Taken from living human patients who suffer from inflammatory bowel disease, conditions like Crohn's disease and ulcerative colitis.
And the match was definitive.
It was an exact alignment. The specific inflammatory genes like LCN2 that lit up in the laboratory organoids were the exact same genes aggressively overexpressed in the diseased intestines of the real human patients.
That is incredible.
The organoids weren't just mimicking a generic stress response. They were perfectly simulating the exact molecular pathology of human inflammatory bowel disease.
And they replicated this success in the respiratory models as well. They generated lung organoids using cells derived from patients suffering from COPD, chronic obstructive pulmonary disease.
Right.
When they mapped those diseased organoids against the healthy HEOCA baseline, the algorithm instantly highlighted the exact cellular deviations and compromised club cells responsible for the patient's breathing issues.
It completely validates the fidelity of the platform. We are no longer guessing if our models are accurate. We can quantitatively prove that a lab-grown organoid perfectly mirrors the complex disease state of a living human.
Okay, final recap before we wrap up. Point 1. The HEOCA isn't just a map, it is a baseline to measure disease. Point 2, organoids react to viral and inflammatory attacks by turning on specific defensive genes like the self-eating autophagy process. And point 3, these lab-created reactions match real-world patient data perfectly, like IBD and COPD.
Exactly right.
So if we can simulate IBD perfectly in a dish, what does this mean for the future of clinical trials and testing new drugs?
While synthesizing everything we have unpacked today, the HEOCA effectively cures the Wild West of organoid research.
Yeah.
We have a centralized, algorithmically harmonized database of nearly 1 million cells. Researchers anywhere in the world can now sequence their models, run them through the Sapoli algorithm, and instantly verify their fidelity.
No more guessing.
None. They know if they need to use adult stem cells for immediate accuracy, or if they need to utilize xenografting to push pluripotent cells to maturity. And crucially, they can induce perfect, clinically accurate disease states in these tissues. The implications for pharmacology and translational medicine are just massive.
Because we can skip the animal testing.
Exactly. Historically, developing a new treatment for IBD or COPD requires years of testing on animal models, which are incredibly expensive, ethically complex, and they frequently fail to translate to human biology. Mice are not humans.
Right.
Their immune systems react differently. But with this atlas, we bypass the animal model entirely. We can test novel, highly experimental drugs directly on human tissue that possesses the exact genetic signature of the target disease.
It accelerates the pipeline for life-saving treatments while fundamentally increasing safety. We started this deep dive by talking about a universal map of the body, but what this paper really represents is the modularization of human biology. We now have the blueprint to take stem cells, grow them into highly specific miniature organs, map their exact genetic state against a global baseline, and infect them to mirror devastating human diseases with perfect precision.
It is the ultimate sandbox for human medicine.
If you think about the trajectory of your own healthcare, this is where it gets incredibly personal. We are rapidly approaching a reality where medicine is no longer trial and error on your actual body. Imagine walking into a clinic with a complex, mysterious, inflammatory condition. Instead of prescribing a drug and waiting 6 months to see if it damages your liver or heals your gut, a doctor sequences your cells.
Right.
They grow your specific, unique mini organs in a lab. They map them against the HEOCA database. And then they test 10 different experimental, highly toxic cures on your lab-grown tissue. They can watch exactly how your unique genetic barcode reacts before you ever swallow a single pill.
It is a game changer.
Think about how that map changes the future of your own healthcare. Thank you for taking this deep dive with us into the vanguard of cellular biology. Catch you on the next one.